DNA, or deoxyribonucleic acid, is the molecule that encodes all of life. Each DNA molecule is a double-stranded helix made up of nucleotide bases, including adenine, cytosine, guanine, and thymine strung together. A specific sequence of nucleotides codes for the genes that are expressed in an individual's phenotype. In this video, we will discuss the structure of DNA and how sexual reproduction passes genetic information through generations. Finally, we will present a case study where a group of researchers discovered the origin of a rare strain of HIV by applying the concepts we discussed. DNA is a biopolymer consisting of long strands of nucleotide monomers. There are 4 types of nucleotide bases: adenine, thymine, guanine, and cytosine. Adenine and thymine are complementary, and so are guanine and cytosine. Long sequences of nucleotide bases are read by RNA, which translates 3 nucleotide base codons into amino acid sequences for proteins. Essentially, RNA translates genetic information into phenotypic information. Have you ever wondered why members of a family seem to have similar physical traits? Their similar phenotypes are indeed related to similarities in their genotype. In fact, the nature of sexual reproduction ensures that parents will pass some of their genetic information to their offspring. Each parent donates a haploid gamete that contains a randomly assorted half of their genes. 2 haploid gametes, one from each parent, combine to form one diploid zygote containing both parents' DNA. Thus, every child is related to their parents and ancestors by similarities in their DNA patterns. Furthermore, analyzing DNA samples can reveal clues about ancestry and help put together phylogenetic trees.
Let's take a look at a case study where Martine Peters, a virologist at the Institute of Research for Development and the University of Montpellier in France, and her colleagues studied the origins of a rare type of HIV. Their goal is to find and explain the origins of HIV-1, which consisted of 4 groups: Group M, N, O, and P. Group N is the main phylogenetic group, which has infected more than 40 million people and accounts for 90% of the cases of HIV. N and P are found much less often, with the total number of infections being around 25 combined. O has the potential to cause another major epidemic of the disease, with it having infected roughly 100,000 people, primarily in Central Africa. As for the origin of these strains, Dr. Peters and her colleagues traced Group M to a similar strain of simian immunodeficiency virus, or SIV disease, in chimpanzees. They also found a strain from the same population for apes that gave rise to Group N. The next step was to find the origins of Group O and P. To start off the search, the group collected over 3,000 fecal matter samples and extracted viral genes to search for specific SIV-related antibodies and nucleic acids. From this initial search, the team found a small group of gorillas in the western lowlands of southern Cameroon that had these components. They put their data together and constructed the phylogenetic trees shown here by making family connections based on sequencing partial gene fragments. Then they color-coded the tree based on who had which strains of HIV and concluded that gorillas contracted SIV on one single occasion from chimpanzees. Furthermore, they determined that humans contracted HIV from gorillas on 2 separate occasions, accounting for groups O and P. As for why groups N, O, and P are so much rarer than group M, the researchers hypothesized that M was much more successful in spreading due to a carrier who traveled to a rapidly developing city in the Congo.
By combining the studies of genetics and evolution, researchers were able to uncover the origins of 4 different types of HIV. They used advanced gene sequencing techniques to analyze the DNA of simian fecal samples and connect individuals into a phylogenetic tree where the last common ancestor represented the origin of the virus. Understanding the origins of HIV gives us valuable knowledge about how to control this deadly virus. Also, it can provide us with significant information on how to recognize possible similar diseases and how to prevent them from ever reaching mankind.